travel through the fascinating world of discoveries and inventions in science and technology related to biology. Our journey begins in the 17th century. In 1665, Robert Hooke improved the design of the existing compound microscope. His microscope used 3 lenses and a stage light, illuminating and enlarging the specimen. This advancement allowed Hooke to see something wondrous when he placed a piece of cork under the microscope. Year 1674, Anthony van Leeuwenhoek is known as the father of microbiology due to his early study of bacteria, and is also regarded as the father of microscope, where he discovered the protozoa called animal cells.
Now, on to the next journey, the 18th century. In 1753, Carolus Linnaeus introduced the binomial nomenclature system for classifying and naming species. Then, in 1796, Edward Jenner introduced the first vaccine. He developed a vaccine against smallpox by using another disease called cowpox.
We are now in the 19th century, starting in the year 1838 with the cell theory, proposed by Theodor Schwann and Matthias Schleiden, in which all living things are made up of cells. Cells are the basic unit of life, and all existing cells are produced by living cells. So, 1860, Joseph Lister was the first to use antiseptic in surgery and developed the antiseptic technique. He also proved that microbes cause surgical infections. 1865, Gregor Mendel proposed the fundamental law of inheritance, which means that all genes come in pairs and are inherited from each parent. He also considered as the father of genetics. And in 1890, Sergei Winogradsky is the known for contributing to soil microbiology.
Moving on to the 20th century, Alexander Fleming discovered the penicillin in 1928. This led to the introduction of the antibiotic, which significantly reduced the amount of deaths due to infections. Then in 1945, salmonpox was discovered streptomycin, the first effective treatment for tuberculosis. This marked the beginning of a new era for antibiotic discovery. In 1945, Howard Florey, Ernst Boris Chain, and Norman Hickey the mass production of penicillin. The development and industrialization of penicillin for pharmaceutical use was greatly aided by Florey, Chain, and Hickey and their team, who made penicillin widely accessible for medical use during World War II. Their team required developing effective techniques for generating it on an enormous scale.
Year 1953, James Watson and Francis Crick discovered the structure of DNA, the molecule that carries genetic information. This is a significant breakdown in biochemistry and is opened the door to understanding the genetic past of life and disease. 1961, Francois Jacob and J.P. Namo explained how genes are regulated in bacteria. French researcher Francois Jacob and Jacques Monod divide the operon model. A better knowledge of bacterial gene expressions and regulations resulted from their work, which demonstrated how genes may be turned on or off in response to environmental challenges.
Moving on, in 1977, Frederick Sanger, a British biologist, created DNA sequencing techniques. His methods greatly aided the advancement of genomics and microbiology research by enabling students to unravel the nucleotide sequences in DNA. In 1988, Curry Mullis invents the polymerase chain response, or PCR, a technique that allows the amplification of DNA fragments. This is a major discovery in genetics that has critical applications in medicine, agriculture, was a turning point in genomics, allowing for the decoding of complete microbial genomes and DNA.
Congratulations! You made it to our last destination, the 21st century. Let's start with the year 2003. The Human Genome Project is completed, providing a comprehensive map of the human genome. In 2012, CRISPR-Cas9 gene editing technology was developed by Jennifer Doudna and Emanuela Germany. In 2015, the CRISPR-Cas9 gene editing system is used to edit human embryos for the first time.
In 2017, scientists successfully edited the DNA of a human embryo to correct a genetic mutation. In 2020, COVID-19 vaccines are developed at an unprecedented speed using mRNA technology.
This milestone represents the remarkable progress of science and technology in the field of biology. As our world continues to evolve and advance, we can only imagine the exciting discoveries and inventions that lie ahead.
Thank you for joining us in this educational journey, a timeline of discoveries in science and technology related to biology. Always remember, stay curious and keep exploring the wonders of this living world.